Good afternoon. In our laboratories at NYU Abu Dhabi, we study the signaling mechanism involved in various cell death pathways. In today's talk, I'll be discussing about a protein which our lab is very much interested in, prostate apoptosis response 4, in short, PAR4. So what is PAR4? PAR4 is a therapeutically promising tumor suppressor protein encoded by PAWR gene. One of the most striking features of PAR4 is that its ectopic expression or activation induces apoptosis specifically in most of the cancer cells but not in most of the normal cells. PAR4 was discovered by our collaborator Professor Ranganekar at University of Kentucky, and it was discovered as a gene that was activated, as a protein that was activated, induced when the prostate cancer cells were undergoing apoptosis. Even though it was initially discovered in prostate cancer cells, later on it was found to be activated in other cancers when it was undergoing the cell death. So the name prostate doesn't signify that it is only happening in the prostate cancers. So earlier evidence into the tumor suppressive functions of PAR4 came from studies where RAS fibroblast was injected intradermally into the flanks of the nude mice. So when the RAS is injected, it forms solid tumors over the time of 3 to 4 weeks. So RAS fibroblast was injected along with the PAR4 as another set. And from day 6 onwards, the mouse was administered the camptothecin, which is a well-established anti-cancer drug. So if you look at the result here, RAS is causing solid tumor formation over the period of 24 days. However, when PAR4 is expressed along with the RAS without any treatment itself, it is causing significant tumor reduction. Even PAR4 is sensitizing the effect of camptothecin, as you can see here. In another set of study, patients who are submitted to patients who is suffering from hypercholangiocarcinoma, hepatic cholangiocarcinoma, it's a cancer which is growing in the liver. When they are submitted for the surgical resection, the biopsy sample were collected from the tumoral region as well as the peritumoral region, and the PAR4 expression, mRNA expression was checked. So when tested, the PAR4 mRNA levels were significantly lower in the carcinoma sample as compared to the peritumoral region. The same results were observed in the ovarian cancer sample compared to the normal ovary. So how PAR4 is causing the tumor suppression? As I mentioned, it is causing the apoptosis. It is inducing the apoptosis. That is the canonical PAR4 function. We all know that cancer is uncontrolled proliferation of cells resulting from the mutation. So most of the anti-cancer agents act by inducing cell death pathways, mainly a cell death pathway which is known as apoptosis. So apoptosis is a cell death pathway which is regulated by a family of proteases known as caspases. So during the process of apoptosis, caspases gets activated. Once it activated, it cleaves large number of proteins. Usually the proteins that promote the cell death are cleaved and activated, while the proteins that suppress the cell death are cleaved and inactivated. So PAR4 is through caspases causing the apoptosis. That's the canonical PAR4 function. So there are two different modes of apoptosis. One is extrinsic apoptosis pathway that is initiated outside the cell, and then there is intrinsic apoptotic pathway that is initiated inside the cell, inside the mitochondria. So both of these pathways are regulated by the PAR4. And how PAR4 is regulating this mechanism? PAR4 is acting on a number of different targets, both cytosolic and nuclear targets, and as a result, it is activating both extrinsic and intrinsic apoptotic pathways. So this is the summary of those things. PAR4 is acting on different targets in the nucleus and cytosol, and it is activating the extrinsic apoptotic pathway that is initiating outside the cell and intrinsic apoptotic pathway that is initiated inside the mitochondria. So coming to different structural domains of PAR4, PAR4 is having a nuclear localization sequence at the two nuclear localization sequences, NLS1 and NLS2. The nuclear localization 2 is important for the PAR4 function because it facilitates the nuclear entry of PAR4. And PAR4 at the C terminus, it has a leucine zipper domain which enables its interaction with several proteins. The different targets which I mentioned earlier, those proteins interacts with the PAR4 through this leucine zipper domain. And PAR4 also has a protein kinase A phosphorylation sites, which when phosphorylated causes the nuclear translocation of the PAR4. And it has AKT phosphorylation site, which when phosphorylated inactivates the PAR4 by causing the sequestration of PAR4 within the cytosol. It also has casein kinase phosphorylation sites that also inactivate the PAR4. One of the most interesting domain of PAR4 is SAC domain, which is called selective for apoptosis in cancer cells. This domain is important because it was found that only this domain is sufficient for inducing the apoptosis in the cancer cells, not the whole PAR4 is required. Also, it was noticed that this domain induces apoptosis even in cancers which are resistant to the full-length PAR4 mediated apoptosis. One of the breakthrough discovery in PAR4 came when it was identified that PAR4 can be secreted from the cells. Once it is secreted, the secreted PAR4 can induce paracrine apoptosis function in the even surrounding or even in the distant cells. The earlier findings leading to this discovery came from a study in which PC3 prostate cancer cells were subcutaneously implanted into the nude mice, flanks of the nude mice, both flanks actually, and tumor was allowed to develop. Once the tumor was developed, then one side of the mice adenoviral PAR4 construct was injected and another side control vector was injected. And after 21 days, the tumor was measured. If you look at this result, when the region where adenoviral construct was injected, the tumor was significantly grown, while there is no tumor grown in the region where the PAR4 was injected. Actually, this happened only with 50% transduction efficiency, but the tumor suppression is nearly 95% tumor suppression was seen. So it indicates that it is not only the transduced cells that is undergoing the cell death, it is even the surrounding cells where the PAR4 is not present is also undergoing cell death, indicating that PAR4 has function outside the cell. To prove this, prostate cancer cells were transfected with the GFP PAR4 and SAC PAR4 constructs, and apoptosis was measured by immunohistochemistry for caspase-3. If you look at here, there is very limited PAR4 transfection, also SAC transfection. However, if you look at the apoptosis, even the cells which were not transfected with the PAR4 is undergoing apoptosis, indicating that PAR4 may be secreted from the cells into the media, and that PAR4 causing the apoptosis in the nearby cells. Now, to prove this further, the media was separated from the cell, whole cell lysates, whole cell extract, and subjected the Western blot for the media to see whether PAR4 is present in the media. So the PAR4 is present in the whole cell extract, which is very much expected, but interestingly, the PAR4 is also released into the media. Now, to see if this released PAR4 is indeed responsible for the cell death in the cells, the media was collected, conditioned media was collected, and it was neutralized with antibodies because it's a GFP PAR4. So if we add GFP antibody or PAR4 antibody, that will neutralize the PAR4 protein in the media, right? So when neutralized with the GFP or PAR4 antibody, if you look at here, then okay, after the neutralizing, the media was added into another set of PC3 cells to see if the media is capable of causing the cell death. If you look at this result, the media which is containing the PAR4 is causing the significant levels of apoptosis, nearly 40% apoptosis is there. But when neutralized with the GFP or PAR4 antibody, it is causing the protection from the cell death. P10 is used as a negative control here. Same result is obtained from the serum sample where the transgenic mice expressing the PAR4, the serum is isolated, and from the serum it is neutralized with the PAR4 antibody and GFP antibody. We have the we can see the same result here. Coming to summary of this finding, there are so many things, but directly coming to the summary of these findings, PAR4 is secreted through classical Golgi to ER pathway, classical ER to Golgi secretory pathway. And secondly, PAR4 is causing the plasma membrane translocation of the GRP78. Once it is in the plasma membrane, PAR4 is binding to GRP78 as a receptor, and once it is bound, it is activating the apoptosis pathway. Now coming to, as I mentioned that during the apoptosis, caspase cleaves so many different proteins. We found that during the process of apoptosis, caspase is also cleaving our protein of interest, which is PAR4. So we were studying the tumor suppressive functions of sphingolipid, sphingosine, in leukemic cells. We noticed that sphingosine is causing caspase activation, which is indicating of apoptosis in leukemic cells, as evidenced by caspase activation and PARP cleavage. When we use an antibody which is raised against the C-terminal fragment of PAR4, we notice that along with the expected band, we notice another band appeared below. With increasing concentration of the sphingosine, the band intensity also increased. Now, as I mentioned, caspase cleaves so many proteins. We already know that. So to check if caspase is responsible for the PAR4 cleavage, we use pan-caspase inhibitor ZWAD-FMK. We inhibited the caspase and see if this inhibition protect the cell from protect the PAR4 cleavage. So as expected, with increasing concentration of the pan-caspase inhibitor, PAR4 cleavage is also protected. There is no PAR4 cleavage with near 55 micromolar concentration of ZWAD-FMK. We have also carried out in vitro caspase cleavage assay, where the whole cell extract was incubated with the caspase 3 enzyme separately outside the cell, and there is a clear PAR4 cleavage is there. However, when we use the ACDVD-CHO, which is inhibitor for the caspase 3, the cleavage is not happening. Now, as I mentioned, we use the PAR4 antibody raised against the C-terminal fragment of PAR4. So, in order to get a PAR4 cleaved fragment of nearly 21 kDa, the cleavage must happen somewhere here. So we did site-directed mutagenesis to mutate the possible cleavage, caspase cleavage sites, and we transfected HEK cells with the plasmid which is expressing those mutant proteins, and whole cell lysates were incubated with the caspase 3 again, and PAR4 cleavage was analyzed. So interestingly, the 131A, the mutant which is D131A, which is inhibiting the, it's not working. You have another one? Okay. So the mutant 131A is inhibiting the PAR4 cleavage. Also, while using the in vivo experiment using the curcumin as an apoptosis inducing agent, the cleavage is disappeared in that case also. So we have identified a new site in the PAR4, which is caspase cleavage site, that is D131 region. So as a result of cleavage, it is generating a PAR4 amino terminal fragment and cleaved PAR4. Later on, others have found that PAR4 amino terminal fragment also has an important function in the cell, which I am not going into. So, PAR4 in autophagy. We know that PAR4 is causing the apoptosis. PAR4 is important for, PAR4 plays an important role in inducing the apoptosis. But not all the cancer cells are sensitive to apoptosis. There are so many cancers which are resistant to apoptosis. So though in those cancer cells, we may have to try other alternative cell death approaches, like we may have to try oxytocin, which is reactive oxygen mediated cell death. We may have to try the ferroptosis, that is iron mediated cell death. Then there is autophagy, then there is senescence. So we found that PAR4 is not only causing the apoptosis cell death, PAR4 can also facilitate the autophagy mediated cell death and senescence mediated tumor suppression also. We have also found that PAR4 is important for the ferroptosis mediated tumor suppression, which is already awaiting for the publication. We have already submitted the paper. And PAR4 is also important for the oxytocin, which is currently underway. The project is currently underway. So what is autophagy? Autophagy is a tumor suppressive mechanism in which when in response to stress, the degrade organelles or the proteins, which is unfolded proteins, which are taken into a double membrane vesicle called as autophagosomes. And this autophagosomes goes and fuses with the lysosomes. And the content inside the autophagosomes are degraded by the lysosomal hydrolase, and the cell undergo into, the cell will die as a result of this. So, two important hallmark of autophagy is the LC32 induction, where the LC3 protein, which is forming the autophagosome membrane, that will get induced, and P62 degradation is a protein that carries the cargo inside the autophagosome that degrades along with the cargo. So these are the two important hallmarks of autophagy. So you can see that when we treated the cells with the U87G glioma cell, malignant glioma cells with ceramide, you can see that LC32 is induced and P62 is reduced. We have also noticed that PAR4 is induced during this period. Now, to check whether PAR4 has any role in the autophagy, we knock down PAR4 using siRNA. When the basal knockdown of PAR4 itself is causing the LC32 reduction, indicating that PAR4 knockdown is protecting the autophagy mediated tumor suppression. Even the ceramide mediated LC3 induction is also suppressed by the PAR4 knockdown. And the cell death is also protected. Now we overexpress the PAR4 using DDK PAR4, and if you see this, when the PAR4 is overexpressed, the basal expression itself is causing the LC32 induction, and even the ceramide mediated the LC32 induction is increased while the PAR4 is overexpressed. Same with the P62 degradation and cell death. So there we used ceramide. In this case, we have used another agent, arsenic trioxide, which is another well-established autophagy inducer. And here we used TET-inducible PAR4 construct, where when we add doxycycline or tetracycline, the PAR4 will be activated. So in the presence of doxycycline, the PAR4 is induced, as you can see here. Similarly, LC32 is induced, significantly induced, and P62 is reduced. And we have checked that this is not only happening in the malignant glioma cells. We have used other two cell lines, and the similar results we have seen. So indicating that PAR4 is not only causing the apoptotic cell death, but it can also play a role in other modes of cell death such as autophagy. Now coming into senescence, we have also noticed that PAR4 is causing the senescence mediated tumor suppression. Because of the time constraint, I don't think I will be discussing that here in detail. Just going through a summary, because thymoquinone we have used here, and it is causing the significant tumor reduction, and you can see the PAR4 induction is the same result here. When we use the tetracycline inducible PAR4, it is causing the increase in the beta-galactosidase staining, which is indicating the senescence. And when we inhibited the PAR4 using SH-PAR4, there is significant reduction in the beta-galactosidase staining, which is indicating the senescence. We have also confirmed this with the cell cycle analysis. Now coming into the final portion, PAR4 into cancer therapeutics. So one of the clinical oncology, one of the research into clinical oncology is mainly trying to identify agents that can efficiently induce the cancer cells. This is the main approach that has been used for last few decades. So one of the main problem with these anti-cancer agents is that along with the killing the cancer cells, these agents also kill the normal cells, which is called as adverse drug reaction. So PAR4 has an advantage here because it only affects on the cancer cell, mostly in cancer cells. Targeting PAR4 is a promising approach where we can selectively target the cancer cells. How we can target the PAR4? We could try to re-establish the cytotoxic function of PAR4 using the AKT inhibitors or BCL2 inhibitors. These are, I mentioned that these proteins can negatively regulate the PAR4 function. If we inhibit these proteins, we can sensitize the PAR4 function. Or we can use some of the PAR4 inducers. We have, in our lab, we have seen that if you treat with vitaferin A or sodium selenate, independent of the cancer cell line, all the cell line, it is causing the significant PAR4 induction. So we could use these agents as PAR4 inducers to sensitize the cell death. Or there are alternative approaches which have been trying PAR4 nanoliposomes or plant-derived PAR4 SAC. One of the approach that has already into clinical trial for the anti-cancer therapy is the PAR4 secretagogue. These are the agents which can enhance the PAR4 secretion. So coming to this observation, in this study, the transgenic mice which is expressing GFP or SAC were injected with Lewis lung carcinoma cells. And after 28 days, the tumor size was measured. So if you look at this result here, in the GFP transfected control mice, all the mice develop tumors. In the GFP transfected mice also, all those develop tumors. However, in the transgenic mice which is expressing the SAC, only 3 out of the 20 mice develop tumors. Same here, the tumor volume was significantly lower in SAC transgenic mice. So they have screened large number of agents, large number of library to identify agents that could facilitate the PAR4 secretion, and they have identified that anti-malarial agent chloroquine is an effective PAR4 secretagogue. So to check this, MEF cells were treated with chloroquine and lysates, and the media was separated, and check whether it is causing the PAR4 secretion. So in the media, chloroquine treatment is causing significant PAR4 secretion. Also, mice treated with chloroquine, when the serum was analyzed, there is significant PAR4 induction. Similarly, renal cell carcinoma patients from chloroquine clinical trial, when observed pre-chloroquine treatment and post-chloroquine treatment, there is significant PAR4 induction post-chloroquine treatment. So in this study, Lewis lung carcinoma cells, which is having luciferase activity, is transfected into PAR4++ and PAR4- mice, and after 24 hours, the mice were subjected to chloroquine treatment and post for 5 days. Okay, 24 hours post to the final dose, the PAR4 activity was not measured. So there is significant PAR4 induction in the PAR4++ mice, but not in the PAR4 knockout mice. Similarly, the mice were again injected with luciferin and checked the tumor growth. In the wild type, when the chloroquine was given, there is no tumor growth at all. However, in the PAR4 knockout mice, there is significant tumor growth even in the presence of chloroquine. And this is the tumor nodule formation. And thank you everyone. Thank you, Professor Samuddin. Thank you, lab members and CTP, CGSB for their support.